Hello, Shubha. You can start actually.
Sure. Hello, everyone. Welcome to another session with NIBM and Bioinformatics on a comparative and subtractive genomic approach for shortlisting potential drug targets in multi-drug resistant bacteria by Dr. Prachi. Let me give a short introduction about her. Dr. Prachi have a great done her PhD in bioinformatics and have a strong experience in biology, clinical pharmacology, biomarker discovery using omics data. She completed her MTech bioinformatics from Karunya University and she completed her PhD from Sri Ram Swarupa Memorial University. Thank you, ma'am, for accepting our invitation. In this presentation, we will explore the comparative genomic strategy for progressively narrowing down a large pool of therapeutic candidates in accordance with criteria of essentially selectivity and drugability. And dear participants, make use of to be make the session to be more interactive, and questions and feedback link will be addressed at the end of the session, and certificate will be issued in 1 week followed by the feedback. And over to you, ma'am. Ma'am, I think you are in mute.
Now? Yeah.
Yeah, it's
Okay.
audible.
Thank you so much, Shubha.
You're welcome, ma'am.
Hello, everyone. So I'm Prachi, and a beautiful explanation about me has already been given by Shubha. So I'll not spend any more time about myself. Only thing is, like I have finished my PhD recently from Sri Ram Sarup Memorial University, and my PhD topic was drug target identification and interaction studies in arthritis and periodontal bacteria. So today I'm here to talk about a comparative and subtractive genomic approach for shortlisting potential drug targets in multi-drug resistant bacteria. So this study we are talking about bacteria, but this study can be applied to any microorganism and it can be studied in various different host organisms. Okay, so first let us understand why there is a need for this study and then we'll go into the methodology. So today I'll be talking more about the methodologies we can use this approach and we'll understand various tools and the databases. So this is it's a comprehensive talk about the various approaches we can do with similar sequences and non-homologous sequences. My screen is visible, presentation.
Yes, ma'am.
Yes.
Okay. So today I'll be talking the introduction about the topic and then we'll see the objective, the properties of the drug target, methodologies, applications, scopes and limitations. Okay. So first let us understand the drug resistance in the bacteria. So we have already successfully treated a lot of infectious diseases and COVID has already explained the significance of using computational approaches for identifying various targets and biomarkers and find potential targets using the computational approach. So we have already succeeded in fighting against the bacteria in a broad spectrum, but still there is lot of upcoming of already eradicated diseases, and that is due to the drug resistance. So a lot of bacteria has learned and evolved to fight against the antibiotics we are using, because we are using antibiotics very frequently and we are not having a narrow spectrum of using a particular antibiotics for a specific disease. The bacteria is exposed too much to the antibiotics and they have learned to evolve and protect themselves from the antibiotics. So there the need for new target has evolved so that we can develop new antibiotics and uh, fight against the existing diseases. So this comparative and subtractive genomic approach is being used in from a very long time, but it is still very efficient tool when it comes to computational studies. So this there is a lot of variation has been done when it comes to comparative and subtractive genomic approach. It is very cost effective, and I am taking this topic here today because a lot of undergraduate, postgraduate and diploma student, they look for a small project which they can do with minimum requirement of the computer and minimum requirement of the internet speed. So this is one project. It can be done at very basic tools and very basic computer facilities. And since this is very much related to the microorganism, we have n number of microorganisms available, so the possibility of similar type of study in similar bacteria is very less. So this study is very significant when it comes to host pathogen interaction, whether it is animal, plant kingdom, virus or any form. So this study is a very wide study. Today I'll be talking about the bacteria, but it can be replicated into any pathogen. Okay, so what first we understood that there is a need for new target. So now what is the aim of this work? So first thing is we have to use already existing data. So now we have a huge amount of sequence data already available. So we don't need to generate a new data, and this study doesn't require a very huge amount of data to be stored in your system. It requires very basic sequence datas. Okay, so here first thing is we are going to use the existing data, and then we are going to reduce the number of targets, I mean number of proteins which need to be studied in wet lab. So the whole intention about this computational study is to reduce the number of proteins which has to be studied in the wet lab. So it reduces the cost of the reagents, it reduces the time. So we will prioritize the drugs which has to go into the lab, and this selection will be done on the basis of essential things bacteria needs, the selectivity and how druggable it is. Okay, then we will look for those genes which are not similar in the host. We will find out non-host genes. So host can be anything, plant, animal, and we will use this method. From this method we are going to understand the unique metabolism and the processes which are available in the bacteria, and we will also find out understand uh, uh, how to filter which are the protein which can be used as a drug target and which cannot be used as a drug target. And most important thing about selective subtractive and subtractive and comparative approaches, we are trying to filter all those proteins which has some similarity with the host, so this the side effect of the drug will be reduced. So these are this study has been done in lot of microorganisms. This is just one screenshot of the microorganism, but there is a huge number of microorganisms on which this study has been done. And still till 2022, the papers have been published which has used this particular approach. So in bioinformatics, it is very important that when we are going to use any tool, we have to look for the papers which are recently published, because the database has been updated very frequently, and the tools are updated very frequently. So many tools which are used in old papers may not be working now. Okay, so for that reason, we always look for the recent papers, the tools also. So when we are looking for a drug target, before we go into methodology, few things we should always keep in the mind. First thing is what we are looking in a drug target. So it should be a virulent factor. If it is a virulent factor, it is more significant protein to be target for any drug. Second thing is it has a good interaction capacity. So it should be our, whenever we are injecting any type of drug or providing any type of drug, it should be able to interact. Then it should not be homologous to the good bacteria. Okay, so we have a huge amount of bacteria which are very beneficial for our body. So these antibiotics which are randomly used or which are not specific to a pathogen, they disturb the natural microbiome also. So there only the problem of side effect arises. So we have to make sure that these proteins which we are trying to target should not be homologous to the good friendly bacteria. Then this should be it will be more beneficial if we are targeting an enzyme, because already the bacterial genome is very small and they will be having fewer enzymes which are very essential for their basic metabolism. So if we are able to target some of the enzyme, that will be even more better. Then we also need to make sure that there is no cross resistance developed. There should not be protein which is used by some other medications and develop a cross resistance there. Then we also have to make sure that these proteins are in accessible location. So we have to understand their subcellular localization. And a final thing we have to understand is we should be able to extract this protein for the wet lab studies. So these are the few points we should always remember when we are looking for a drug target. So this comparative and subtractive metabolic approach is done in a many ways. I will come to that in few next slide, but I will explain one basic concept here and then we will see the variation. So first thing is, what is comparative and subtractive genomics? So we are going to compare the genomics of a pathogen with the host, and we will try to identify only those uh, pathways, genes, or proteins which are unique to the pathogen, that is not found in the host organism. So that will help us to reduce the side effect of the drug. So this way we are going to make a filter. We will apply many filters to reduce the number of target which has to go for the final experimental studies. So we are going to, for this particular figure, we have taken the metabolic pathways of a oral pathogen, that is Perforomnas gingivalis, and we are trying to compare with the host metabolic pathways. So here we have made a very specific study that we want to look only for the metabolic pathways. From there we will figure out only those pathways which are very unique to the bacteria. They do not have a similarity with the Homo sapiens, so that we will avoid the drug to go and interfere with the host metabolism. Then I looked only for the enzymes, not for all the protein. Overall proteome data I did not look for this study. So I only looked for the enzymes. So from these unique pathways, I have extracted all the enzymes. So when we look for these enzymes, we also need to remove the duplicates. So many enzymes will be working in different pathways together. So we have to, it will be when we extract the data from the pathways directly, so we will have the repetition of the enzymes or the proteins. So we have to remove the duplicates. And in this study, I have not looked for the gut microbiome and oral microbiome. I've just to explain here, I have reduced the steps here. Okay, so what we are doing, once we have the enzymes which we want to study and see how druggable they are or how good the target we are, we will extract these enzyme sequences. So sequences can be extracted from various databases, but we prefer using only the NCBI database because that has a good annotated sequences. We also prefer using only the RefSeq sequences because they have manual validation. So there is a very less chance that the sequence can go wrong. So there we will use the sequence data for the selected enzymes. So now these enzymes are from the unique pathways of the bacteria. Now what we have to do, even though we are taking the enzymes from the unique pathway, there is a possibility that that enzyme might be homologous to the Homo sapiens. So what we will do is, first we are going to find the similarity between the sequences, the enzyme sequence, and the whole host, the Homo sapien sequences, whole genome similarity search we will do. So for that we are going to use a tool called BLAST. So BLAST is a tool which help us to find similarity between the two sequences. So in this particular tool, what is our aim is to find those sequences which are not similar to the host. So for that we will use two basic parameters of the to filter the genes, to filter the proteins. We will put two simple filters. So first we will look for e values. So when we talk about BLAST, e value tells us how similar the two sequences are. So here the e value should be always towards the 0, so that they say that indicate that your two sequences are similar. But in our for our study we need a non-homologous sequence, so sequence should not be similar to the host. So we will be looking for the values where the e value is higher, not the lower side. Okay, so from here we will get all the sequences which are non-homologous to the Homo sapiens. We will also look for the identity, which has to be like 35%, should not be higher than the 35%. And we will take all the sequences from there. So suppose we are having 40 enzymes. Now after BLAST we'll be having some 20 enzymes which are non-hit, or we will get only 10 enzymes. So this is the way we are going to prioritize our target. So we are narrowing down the number of target which can be used as a potential drug target. So now we have all the non-homologous sequences here. And now what we have to do is, we got the sequences. Now we have to understand, are these sequences very essential for the bacterial survival, or are they having a good virulence factor? So we have to understand that, because we cannot just target any sequence or any protein, I mean. So it has to play a significant role in the bacteria. So now all these homologous sequence, we will find the similarity in the database of essential genes. So this DEG database, it contains information about the essential proteins which are important for the survival of a bacteria or any microorganism. So in this database, they sort the sequences based on the conserved regions. So if the conserved regions are high for particular sequence, we say that that particular protein is essential. And when the conserved sequences are less, we say in a overall terms that sequence is not so essential for the bacteria. So all the non-homologous sequences, again we will BLAST with the DEG databases. So here in this whole approach, we only need the sequence, and based on the similarity and non-homologous, based on the similarity, we will filter out all our protein sequences. Okay, so that is why I explained that this is a methodology which can be used by anybody, and it's a very simple steps, but it is very efficient technology when it comes to understanding a significant protein in any bacteria. Okay, so now we have all the homologous proteins. We do a BLAST, again the DEG database. So in this DEG database, now we want sequences which are more similar to the conserve reasons. So our E value has to be lower. So we will look for the lower E values, and we will extract all those sequences from, suppose we had 20 non-homologous sequences. If we do BLAST against the DEG databases, we will get maybe 10 or maybe 15 protein sequences which are having good similarity with the conserved regions. So from there, we will take these 15 proteins and go ahead to understand what is the location of these proteins. Or we also have to understand that, are these proteins already used for some other drug or not used, or they are noble. So these two parameters are very important when we are saying any protein as an important and a significant drug target. Okay, so first we will talk about the subcellular localization, and then we will go to the drug target. So in the subcellular localization, what we have to make sure is this protein which we want to study, how accessible it is when we are subjecting a drug for any type of disease. So the protein has to, the drug has to reach the target to do it works. So in general, the subcellular localization mainly are preferable ones are the cytoplasmic ones and the membrane proteins. So we will look what is the location of that particular protein, whether it is accessible or not. So here in this study I have used a cello tool, but there are many different tools available which can be used for this study. And then after we have understood the location of one of all the proteins, we will see whether these proteins are unique now. So they should not be used by any other drug, or they should not be in any of the studies. So we will go to drug bank. So the drug bank is a database where there is a huge amount of information available about all the existing drug molecules. So there are databases which have interactions that are also listed in the drug bank. So this drug bank, here also we use the blast, but it is an inbuilt blast in the drug bank, so you don't need to run separately the blast. If the number of sequences are less, we can directly go into the database and do the blast study there. So suppose we are having some 10 sequences which we think are essential also, and which are in a proper location also. So they are accessible also. So those 10 proteins we will blast in a drug bank. So this drug bank, what it will give us the information is this sequence are already used or not. So what it will do, it will find the similarity with already existing sequences in the drug bank database. So drug bank database will have certain proteins which are already used by different microorganisms, or which are used by different drugs. In for already known information will be available. So we will try to find out only those sequences which are not having any similarity with the drug bank sequences. Okay, so here again the e-value has to go down, but you don't need to worry when it is a drug bank database, because they have a very beautiful output where directly it will show how whether your protein is being used in any of the preclinical studies or any of the drug. It will also list all the microorganism on which that particular target has been studied. So a lot of information will be shared there. So from there we can pick only those sequences which are uh, new, which are not being used by any of the drug. Okay, so now we have few sequences which are non-homologous, which are essential, and which are accessible, and which are not being used by any of the drugs. So they, once the drug, when once the sequences have come into all these three categories, four categories, we will term them as noble drug targets or potential drug targets. So this is one concept, but this concept can be used in a very different ways. So we'll talk about it, how variation we can bring in this particular study. So there are few things we have to understand when we are doing this study. First is we need sequences. So where we have to retrieve sequence? So I always say go for NCBI database, and there is no when a lot of people struggle to extract total information at once. So there are some ways we can extract the whole information at once. We don't need to go line by line or extract the data. So for that we can use API settings. In each of these databases they have API, so we can directly access the information. For an example, if I want to extract the information from, one second. So I'll just explain how you can use the APIs, and then we will go to the variation. So this is the NCBI. Is my screen visible? The NCBI page?
Yes, ma'am.
Thank you. So this is the NCBI page where you can give your organism name, and we will have information here. So we can extract the ref-seq sequences. We can also we can get all the sequences for protein and the genes, and we have n number of information here. So if you go to the NCBI and directly enter the organism name, there are many information available. So we have to go more specific. So when we are working with the genome, we have to give the genome database. So here we have a concise information which is more about the sequences. So we have the FASTA format of genes and protein. We have information about the RefSec. There is another way we can download this data is from the API. So this is the API of my organism, that is Perforamnas gingivalis. So these APIs are having all the zip files. So from here we can directly download. If I increase the font, so here we have the protein sequences, we have gene sequences. So whatever blast we are doing or whatever sequences we need, we can directly download the zip file and use it as we want. Same thing we can do to extract the data from the KEGG database also. So in the KEGG database, when we go, so here you can enter whatever your organism name, and we will get the IDs here. So when I open this, so we have this information. So these are the information about the database. So when we extract from here, it is a little bit clumsy process. Instead of that, we use different APIs. So in these APIs, it's very simple. We just give a command and it will give us the information. Most important thing when you're using the API is you need to have information about your organism. What are the codes, what are the accession numbers? So whatever is the ID for that particular organism, we have to make sure. So these are easier way to download the sequence data instead of going into the whole database. So here if we go, if I click on the genes, this is a lot of information. Now these are the pathways and genes present in this organism. Okay, so we can download directly from here also. So here what is available for these particular, so these things are, this is my organism. For this, what information I have? So I have pathways, I have a complete information, I have module information, I have KO IDs. This is to identify, this is a specific ID for KEGG. Then we have genome information, enzyme information.
Hello, Dr. Prachi. Can you unmute? Actually, you are in muted, so please unmute.
Yeah. So what has been missed, I'll restart from there.
Yeah,
Am I
there is.
audible now?
Yes, but not much lag, so you can go ahead.
Okay, so these are some APIs. These are the links which we can use to directly extract the information. So these are the ways we can directly download the data instead of going into one and copy pasting from there or extracting too much information. Here we can very specifically download the data. So here we have protein, genome, and assembly details, and we'll have lot of ID. So we can extract the data from here. So these are simple tools which we have to keep in mind when we are doing sequence analysis. So these are some of the APIs. So if we click on these data, we will directly have the information. We don't need to go and search for the databases. So these are the enzyme IDs and these are the pathways. So I can map any of the gene IDs and the pathways. So these are the simple way we can filter our data and have a combination. So we can, this is the enzyme. It will directly give you all the enzymes present in the pathway. So from here, we can just take all the IDs of the gene enzymes. So similar way, there is a beautiful page for the KEGG where you can get all these small codes. You don't need any programming. You just need to understand the URL outputs and inputs. That's all. So these are simple way where you can filter your data. So we saw, so there are some databases. So these are all free online tools. You don't need to go into any of the paid version of this. So first we have sequence, which we can retrieve from the publicly accessible databases. Then the KEG is also free, and we have a lot of information there. Then we can go to database of essential gene. Still, that also has a blast. If you don't know how to do standalone blast, means blast we can download in our system and run the blast. So that is one way to use blast, and also we can run online. So it is more good to use a standalone blast than the online tool, because the online, first of all, the database is updated very frequently, so you may not get the same result every time. So instead of that, you can use the standalone blast. So you can download the sequence and run in your system, so that will give you the similar output every time. Then the second reason we have to use is, if you do not have a very good internet connection, still you can do a very efficient sequence similarity search using the standalone blast. Third thing, uh, it is not very difficult coding when it comes to standalone blast. There are already very good setup, and it is one single line codes are there. So that is very easy. Maybe we can discuss that in some other time. So then there is a drug bank. So drug bank is again a database, and there we can just input our FASTA files, and we can get the output. Then there is a database called cluster of essential genes. So as I talked about the variation, now first I told that we can extract all the essential genes from the database of essential genes. Now we can more specifically work on the sequences by clustering them into groups. Like suppose I am working on enzymes, I can group the enzymes based on their activity, and I can focus on the subset. So I can take out only those enzyme activities which I am looking for in my drug target studies. Okay, so that can be done by using cluster of essential genes. Then we can use virulent factor database. So here in this virulence factor databases, I can it also requires only the FASTA sequence. You just put your FASTA sequence there, and it will give the list of virulence factor which are annotated by various organisms. So I can also look for particular type of virulence I am looking for. So I can narrow down my studies there also. Now we can do subcellular localization studies using different tools. There is a subloc, there is interproscan, then there is psort, and there are number of tools which can be used. So these tools, like I prefer using psort because it is more specific. These algorithms behind the tools are different, so whenever you are selecting a tool, you have to look for the algorithm and understand what is your purpose. So based on that, you can select these tools. Now we have understood what is the requirement. So these are the basic requirement. You can have it with a very minimum internet speed also. So these are some of the databases and their IDs and their information. So what information they have. Okay. Now we will see the variations. So first, we in this study, the first study, the figure 1, we took only those were metabolic pathways. Now this study can be changed into full genome studies. We will not separate metabolic pathways or other pathways. We will take all the pathways, and from there we can take the sequence and continue our studies. We can put filters based on the p values and the e values to filter more sequences. We can make more stringent conditions, so that if we have lot of sequences, we can filter them based on their similarity values also. So we can have different e values. We can take a reference point from various papers, and we can make more strict filters to reduce the number of the targets. Then we can also do homology search with the gut flora, with the oral flora, with the intestine flora. So we will do filter our genes based on their use inside the body also. So suppose we don't want to disturb any of the gut bacteria, so we can do a homology search, and whichever sequences are similar to the gut bacteria, we will eliminate most of them, so that will make our target more specific to the pathogen we are looking for. So in this study, whenever we are looking for a good bacteria or thing, we have to make sure that we the study comes from minimum 30 to 40 bacterial community. Okay, so the similarity should be for that many number of the bacteria. Then only we can say that they should be eliminated. Then we can do drugability analysis, virulence, as I explained in the previous one. So these are the ways we can make more filters. Now this study, what we have done is specific to one strain of the bacteria. Now these studies can be done with n number of strains present in a microorganism, and then we can find first the similarity among the uh, strains and then go for the studies, or we can also do a subtraction study. So we can identify the protein sequences which are not so similar in all the strains or unique in particular strain, and we can also study about its drugabilities. Okay, so same this is one variation. So one, we can take the whole genome. Another variation we can do is we can also study the pathways at last. In the previous study, we have taken first the pathways and then extracted the genes. In the other studies, we can take all the genes and then we can do the functional analysis later. So when we are looking for whole genome, we can choose for those type of bacteria for which the functional annotation is not available. The functional annotation is not available in huge amount. So that moment we will not go with the pathway studies. We will first take the genes, and then we will at last we can filter them based on their pathways or based on their functional annotation. So this is another type of similar. It is, we are doing the subtractive and the comparative genomics only, but the approach is different. So this study is also used for designing the designing the vaccine, and the reverse vaccination methodology uses this approach to remove all the paralogs. So this, here also we can go with one sequence, we can go with the whole genome sequences, and we can look for our particular targets. So if you look at this study in the figure 4, they have first looked for the subcellular localization, then they have done the rest of the studies. Okay, so why we have to, when we are designing a vaccination, a subcellular localization is very important, because the vaccine is different from the drugs, the other physical drugs, the capsules and other way. So this time we have to the vaccine, when we are designing it, the location of the target plays a very significant role. So we are first filtering all the genes based on the subcellular localization, and then we are doing the rest of the studies. Now this, if you look at this figure 5, they have done the same study in a little bit different ways. They have used different tools, and what they have done here is they have taken a complete proteome database only. So you can also choose coding sequences, you can also choose non-coding sequences, and you can make your filters. So each study is different. It's not that you can randomly choose one of the method, but like if you are looking to study more hypothetical protein and do some analysis with the hypothetical protein, you'll go with a whole genome database. But when you are looking for very well studied bacteria and looking for a narrowing the targets, you will always look for what functional annotation is available. So for that, we will always study first the pathways, and then we will go to the gene and protein level. So what is the intention of your work? This study can be uh, changed in different ways. So in the figure 6, if you look, they have taken 52 strains of the bacteria, and they have done a CD hit. So first they have identified the clusters, and then they have done rest of the studies. So these are small, these tools are like you can rearrange these tools according to your need. So just one concept and it's been used in various studies. Now, this is also one type of study where they have done the docking studies. So till now I have told how you can rearrange the tool and use the available data. Now, what you can do with the available protein targets. So the targets we have identified. So what all we can do with that. So that will be the application of this approach. So these, like in this paper, they have in the previous paper, this paper they have designed vaccine. In this paper, they are trying to identify they have taken the study further to identify the docking, the ligand which can bind with your target, and they have done the docking studies. So this approach can be taken further in various ways. We can also do interaction studies. Suppose you have got our the final list of potential drug target is very huge. They are not very small, to not very small. In that time we can do interaction studies, so we can make a network out of all the sequences. So there are very different ways we can use our potential drug target. So this is one study where they have done subtractive genomic study to identify the hypothetical proteins. And now these hypothetical proteins, they can use in various ways. So you can do functional annotation. If the structure is not available, you can do homology for developing a new structure or a potential structure. You can do a lot of studies there also. So for whenever we are talking about hypothetical protein, we will always make sure that we start from whole genome approach, not from the pathway approach. Because when we take the pathway approach, we are going to filter a lot of hypothetical proteins. So we will not go with the pathway approach. So I will also wanted to show some of the result, also how it will be when you process these data. So here are some output, like we can convert this study into a very good papers also. So you can present your all the databases, means pathways, or your genome, how many genes are available in which pathways. And we can here in the table one, we can list all which pathway has more number of genes, and you can focus on those particular pathways. Or you can see which pathways are very important for survival, which pathways are very important for multi-drug resistance. Like when we talk about multi-drug resistance, lipopolysaccharides and peptidoglycan synthesis, two component systems, these are very important. They are the place where the bacteria develop itself and develop resistance also. So these are the filter studies. So we are seeing what how will be the localization we will study. So here it will show that what are the cytoplasmic proteins, what are transmembrane proteins, what inner membrane proteins. So we can filter our data like this, and then we can just in the graphical way we can represent. Now this is the drug bank output, where I have done the similarity search with the drug bank, and we can see what are hits and what are non-hits. So hit means you have the similarity, no hit means you don't have the similarity. So based on that I have classified and got to know that there are 3 proteins which are potential drug targets, which are novel, which are unique, and which are very specific to the bacteria. So what are the application? Now we, at the last of this work, we have lot of sequences. Now what to do with the sequences? How your work can be different from the others? So first thing, uh, like you can do all strain analysis, or you can do strain specific analysis. We can also do this study for the novel strains. So if you have a strain from your wet lab and you want to do a study, you just need to convert it into the sequence, do the similarity search, and you can do n number of studies here. So based on that, we can have multiple pathogen. We can do this study for different pathogen, a collection of pathogens for one single host, or single pathogen for collection of host. So this also can be done. Then we can also see whether the protein structures are available for all the proteins we have got or not available. If the structures are available, we can again go for docking studies and other ligand and like potential uh, pharmacological studies also we can do, the dynamics and other stuff. Then we can also predict the active sites. So now the novel drug target we have got, we can see the active sites, so we can do that study also. We can do ligand screening also. Suppose we already have certain drug molecules and we want to see whether it will work on our list of drugs, and we will see whether it is useful for this target or not. We can do ligand screening studies. We can also study the ADMA properties of the drugs based on the sequence similarity we have got, and we can do in silico docking studies. And very important thing we can do is homology modeling and hypothetical protein. So this project, this point why I'm stressing is it is very important for all the graduates and undergraduate to do a small project in their final year semesters. You can take some group of hypothetical proteins from here and you can do different studies, docking or homology modeling, and you can make a good project out of it. Then at an advanced level, we can go with the network, topological studies of the network. So this one methodology, that is my favorite actually. So in this study, if you have more number of, like you have now at last you have got 100 proteins. Now how you will decide which one to prioritize or how to filter those? So for that we do interaction study. So these all 100 protein, we will convert them into the network and try to understand how they are interacting, how they are grouping, what is their value in that network, whether if we can remove them from the network will exist or not exist. So if it is not, the network doesn't exist, then that protein is important, so that can be used as an important drug target. Then we can use it for reverse vaccinology. Okay, so these are few application, but there are so many more variation we can do. So now this study is very good, but still it has some limitation. First of all, the organism which doesn't have the whole genome availability, very difficult to do this study, because if the sequence number is less, we cannot conclude anything. Then it also requires experimental validation when your targets are there. Then third thing is you have to repeat this process regularly to conclude that these are important things, because the databases keep on getting updated, so result may vary every time. So you have to conclude a solid out target, we have to repeat this study for few intervals and then we have to conclude our results. Sorry. Then there are some essential things about the DEG databases, that is it varies a lot very soon. So that time the essential gene category, when we are focusing on essential genes, we have to make sure that we are screening the essential genes very well, not just with the database but also theoretically we are using some of the papers and validating our work. That is very important. Then in this study we don't take the protein which is less than 100 amino acid, so we also lose some of the important protein here. And other thing is some of the protein we filter as non-essential, maybe lack of annotation, lack of uh, complete. So that time also we are missing. So these are some of the limitations, but this limitation is a very beautiful project for all the bioinformaticians, because we can standardize the parameters as a project. So you can study various literatures and you can standardize your blast parameters. We can also develop an integrated platform. So since here we are using different tool one by one, so you can plan a platform where you can bring all the database together. So this can be another a good project for all the people. And there are people who are developing various tools, and still new tools are coming up. So there are some loopholes, some problems which can be sorted and converted into a very good bioinformatics project. And there has to be an independent tool for DEG, because this database is a main filter criteria in this whole study. So when we are eliminating some of the sequences, we have to make sure that they are not they are very well studied in various organisms. So we have to develop the platform where some of the genes which are not very well studied in the bacteria and other bacteria, but very unique in some strains or some particular pathogen, we can develop this tool for those type of sequences. Okay. Then we can also make an integrated bioinformatics based genomic approach, where we can do a hypothetical study for the hypothetical protein. Because at a random we take any bacteria which are uh, present, we will get some 30 to 40% hypothetical protein. So this hypothetical protein studies can be converted into a good tool, where we can find out various hypothetical proteins, and we can classify this hypothetical protein which can be which can be modeled into sequences, which cannot be modeled into sequences, or for which we can do the annotation. So these are small work which can be bioinformatics work, which can be done using this as a background. So these are the references. And thank you so much. So I'm open for the questions.
Actually, thank you for the session, ma'am. I hope the students enjoyed the session and make use of it, because it is reflecting in the chat box. There are so many questions from student side,
Yeah.
and from that, I have picked up almost six questions. And shall we go for question and
Yes.
answer session, ma'am?
Yes.
Okay. The first question is from Sriram. He is asking that if we are using organism specific approach, why we are thinking of non-host genes, and does the data needed is microbe raw data?
The thing is, why we are looking for non-host gene is, whenever we are giving any drug, the drug should be able to identify a bacteria. So if the sequence is similar to the host, then it will get confused. The drug will get confused to disturb the host metabolism or the bacterial metabolism. For that reason, we are looking for the sequences which are unique to the bacteria, not with the which is not found in the host, so that the drug exactly goes and hit in the proper place. And what
Okay.
was the other one? The raw sequences, or see,
Does
the
the diet
thing is, we can do this study with the raw sequences. I mean, raw in the sense, what there are different forms of raw when it comes to in silico studies. So, like if you're taking about the wet lab sequence you have got for a new strain or something, you have to get it first into bioinformatic language, and then we can go ahead with this study.
Okay, ma'am. And the second question is from Jay Krishnan. Can this study alone get published a research article? That he means is this enough to get published work, or further needs to work this work with CAD? Can you share some papers that just published with identification of target only?
Yes, my paper itself has been published, and yes, I can show. I'll just show these are many papers. There are huge number of papers which are published with this approach. There are some papers where they have done only the subtractive genomics, not even gone to the molecular studies and all. But what those paper needed is, whatever protein you are getting, you should be able to explain how it is important, how it is significant. This bioinformatics study will give you a theoretically, it will give you, okay, these are the logically these are the proteins which are important and which can be potential drug target. Now you should be able to identify and establish a connection between the disease and these protein in a better way, that these are the perfect target which can be used for that particular disease. So there are papers, there are a number of papers, and till 22 there are papers which are using this approach. So a lot of people, they use this approach for vaccine designing and for like identifying common proteins among various bacteria and all. So this definitely, if you take a good time to do this work, definitely it can be converted into a good paper. So the whole intention of this talk was, I didn't go for any hands-on, because hands-on is one thing, but you understand what project you want to do, identify the project by yourself is also important. So this is one method which can be translated into many versions. So
And
these are very, there are so many papers actually, which are like recent paper 2019, 2022. So a huge number of papers.
the third question from Anchita Prasad. How can we identify a particular domain is present in pathogenic and non-pathogenic bacteria? Is there any pathway to identify it?
So when we are talking about domain, we can do similarity search with the domain database. So you take your sequence, go to domain search database. There you will, so every domain will also have a sequence. So we can do similarity search with the domain database, and we will get to know the domains, whether similar domains, non-similar domains, or pathogenic and non-pathogenic bacteria. So we can do that study. So we just need the sequences, and we can do the BLAST study with the domain database, and we can identify the domains, common or unique. We can do that.
Okay, ma'am. And the fourth question is from Sanchit Sharma. Do you recommend using NCBI Workbench for BLAST? How to do this homologous study with the different flora? Is there a devoted tool for the same?
Okay. I prefer using standalone blast among any other platform, because it's more convenient, and the thing is we can make our own database. So if you have, suppose 15 or 20 microorganisms, you just download their sequences and we can make our own database. Simple, very simple codes are there, like maybe in a day or 2 day you sit in that, you will get enough number of codes. If you are not having codes, you can just drop a mail to me, I will help you with that. And so these you can make your own, you can group together your organism and make a database, and you can do blast for any sequence. It's not just for this study, you can do this for any. So it's blast, if I show you, like it will look like this, a small, very small. You just need to make a database. It has one single line command to make a database. The thing is, you have to understand the format. To use standalone blast, you just need to understand the format. So to make a database or to do any sequence analysis, you just need a faster file. So if you are able to make the file format, you can do this. So it's just some few lines code, and it will develop your, so it's like that. And it is very simple. You just put your all the files in one folder, and you start your blast from there. It is not like you have to set too many path and all, not at all required. So this is just put your all the things in one folder, and keep make sure that your blast path is to that folder, and done. It's very all the information is available in blast books also. So you can get it from there. You can also use small codes in R, which are very simple. We don't need to go into too much. Just one line codes they have already given, which you can extract information. You can only extract the IDs, so that can be done. Yes, and the flora, like uh, any particular grouping you want to do, you can do on the standalone blast. So you can make your own database and do the blast there. It can be done with any micro number of microorganisms and all. It doesn't take much time also.
Okay, ma'am. And the fifth question is from Gayatri. Can you provide the links of all the drug resistance virulence database.
Yes, we can. I can do. There are only for virulence database. It is just a virulence database only. For what was the other question? The other
The other question
database?
is a drug resistance database.
Drug resistance database. Okay, I will. So yeah, that can be done. I can provide the link. So like I'll check with you and then I'll share it in some virulence database. It is already given in the slide. So here I have already mentioned about the virulence database. So it is here. So this itself is a database. So virulence database. There are resistance information we can also get from essential genes also. And yes, there are databases for specific organism where we have the resistance database also.
Okay, ma'am. And the sixth question is from Harshita. What if there are no hints found in drug bank database?
Yes, we need the sequences only for which there is no hit. No hit means they are the unique sequences. They are not used in any of the drugs. We have to get that hit only. No hit data, no hit sequence only. So see, this concept is like something you have to understand where you want the similar sequence and where you don't want the similar system. You have to make sure you are going right there. So drug bank, we don't want any similarity because we want something unique. So no hit.
Okay, ma'am. And we have covered, I think we have covered all the questions from the chat box because so many students will keep on asking the repetitive question in different manners. And
Yeah.
I hope they enjoyed and they make use of the presentation and the session. Thank you for your time, ma'am, and thank you for your presentation.
Thank you so much for this opportunity. One last thing, I'll take 5 more minutes for all. Whenever
Sure,
you're
ma'am.
doing any sequence analysis, please make sure the data you are downloading and making a file in a proper channel. Okay, so we most of the time we don't organize our input and output data. So whenever you're doing a bioinformatics study, make sure you have a flow, so that flow has to be there. So you have to keep one excel sheet and you keep on going from first to last step should be there in one book. It is very important to do that analysis. Yeah.
Thank you, ma'am. Thank you for the session.